Welcome to Glimpses of Science, an audio series in which we present researchers at the IMP Vienna and showcase their work. The IMP, short for Institute of Molecular Pathology, is a vibrant basic research institution at the heart of the Vienna Biocenter. We are passionate about science and dedicated to answering some of the most fundamental questions in molecular biology. Our 200 researchers from 40 countries are driven by curiosity and determined to meet the highest standards. In this audio portrait, we present structural biologist and IMP-Fellow David Haselbach, who conducts research at the interface of physics, biology, and computer science. Listen to David as he talks about macromolecular machines in action, the power of single-particle cryo-EM, deep learning approaches for computational tools, and the medical relevance of protein degradation.
Our cells are made of a lot of molecular machines that drive all the functions of the cell. They can do very diverse tasks. They can just transport things from one end of the cell to the other one, but they can also do more complex things like synthesize proteins and RNAs or also degrade them. The question is basically, how do they work? How can they achieve that? Because in the end, they're nothing else than a conglomerate of lots of molecules that are inanimate objects, and just by the environmental energy or the energy that is given to them by the heat bath that they are in, they will be enabled to perform all those wonderful tasks. And this gives us basically what is the transition from inanimate matter to a living organism. So this is where we think that life comes about, just from something that is really not alive to something that is alive. And this is a fundamental process. And I think the main question that we are following is, what is the design principles of those machines and how do they actually are designed to use this unusual form of energy. However, this is a little bit in contradiction to what we're seeing in textbooks, because often biology students or students that work with these kinds of machines will hear ATP is the driving force. And just to give you an example, what this means in the end is, so let's assume typical molecular machines consume something like 100 to 1000 ATPs per second. This will translate to a heat power or to a power of 10 to the minus 16 to 10 to the minus 17 watts. However, the heat power, so the power of the environment, is 10 to the minus 8 watts. So this is 8 or 9 magnitudes higher. This is if you're in a thunderstorm, in a very strong thunderstorm, and you try to have a little paper stick against the thunderstorm or against the storm. This is not gonna happen. So the main part, the main energy for the system comes from the environment, and ATP can only provide guidance in this sense. So this will never be the case that the machine is fueled by ATP as a car is fueled by gasoline. It will be rather just that the main movement comes from the heat bath, and every movement comes from the heat bath. However, ATP restricts the backward movement. And this is what we would like to understand. However, this is fundamentally different to a human-built machine will work. And this is what we would like to understand. And with this, we come to conclusions of things that are very unclear in the fields. So if you look for any biological macromolecular machine, you will find like 2-3 functions that have catalytic chemical function of the machine, while there is definitely a lot of more proteins. So what we are working, for instance, is the proteasome that has 3 or 4 different kind of chemical functions. However, we're talking about roughly 40 different proteins. So most of them we cannot give a function to, and they definitely will play a role in how to use the energy from the environment to drive processes. And this is basically what we want to understand. We try to do a good mixture of biophysical and biochemical methods. However, we're focusing on cryo-EM, so single particle cryo-EM, which is a Nobel Prize winning method that got the Nobel Prize for Chemistry in 2017, which enables researchers to get detailed structural insights from large macromolecular complexes without the need to crystallize them, which was the main bottleneck for many structural studies all the years before. How it in general works is we have our purified complexes or purified molecules, and we put them on a surface and freeze them in ice, in a special kind of ice where we're in a glass-like ice, in which we have our molecules completely embedded in a more or less natural state. So this kind of ice will keep them in a hydrated environment that is very close to the environment that we would have in biochemical experiments in liquid state. What we do then is we put our specimen into a transmission electron microscope, and we gain perfect 2D projections of our images, of our molecules. And what we get then is we get distinguishable signal from each individual molecule. So we can actually really distinguish the signal from a single molecule to another one, which gives us a great power compared to other structural methods where we integrate through large crystals where there's lots of copies of the same molecule, where we definitely cannot distinguish whether this molecule is in another state than the other one, which is most likely not is, because otherwise the crystal would not diffract that well. However, we have the great power of distinguishing the signal. And this gave in the last few years great examples of structural information from large molecular complexes. But there is lots of more power to that. So given the fact that we can distinguish individual particles, we can also see if they are different. So if the environment drives them to be in different conformations, we just have to assign this particles in this state and that particles in that state. And there's huge room for improvement still in the technique, because most of the structural studies with cryo-EM these days will have just a single structure or just a few. But actually, if you imagine that the molecule has to fulfill a functional cycle, we have the possibility to find each and every state that the molecule can go through. And to understand that, we develop new computational tools that are first based on standard statistics. However, we will start moving a lot into modern algorithmic approaches like deep learning approaches that have much higher powers in figuring out how those molecules are moving and how to distinguish significantly different signals. However, it's not the only thing that we can do, because if we want to really understand how a molecule works, we have to understand it over time. Because every biological process somehow needs time. If it's the synthesis of a protein, of course, it's first a small protein, in the end it's a large protein, so it's a step-by-step mechanism. And time plays an important role for all of this. So the next step that we want to do is we want to go into time-resolved EM, which people already established in the field a couple of years ago, where we can just do snapshots of individual time points, and from those snapshots actually infer how the molecular machine is actually working and what we can, in the end, figure out about its mechanism. We're studying as one of the examples of the molecular machines the human 26S proteasome, which is one of the major machines that is responsible for degrading proteins that are not needed anymore in the cell. And those proteins can be broken proteins, or those proteins can be giving a signal that is not needed in the cell anymore. This was obviously always a target for drug research, because those signal-giving proteins are also giving signals for cell differentiation, or also for cell division. So obviously the proteasome's function plays an important role in diseases like cancer. But moreover, the proteasome also has a role that its resulting peptides, so it degrades proteins to smaller peptides, are presented by the immune system, or presented to the immune system by the cell. And so the immune system can distinguish individual cells that are maybe having a viral infection or that have a certain reaction that is not natural. So the immune system uses peptides built by the proteasome to distinguish between self and non-self, which is of important clinical relevance. So there are many drugs that are used in this environment, and we worked on some of them that actually stop the proteasome from degrading anything, and with this what you can see is that this will also stop cells from dividing, which is good for treating cancer, but it also will stop cells from presenting antigens on the surface, which will lead to a point where the immune system cannot distinguish those cells anymore, which could be very beneficial for autoimmune diseases. And what we in general are interested in is that all the nature-built molecular machines are not perfect. So there's always cases that the system can be tricked. We can easily do that in an experiment, so we can design proteins that will not be degradable by the proteasome if we make it very unnatural or very tightly folded proteins, or we add covalent bonds in there. However, also nature did that already. So there's a couple of viral strategies that use that. For instance, the herpes virus or the Epstein-Barr virus, they mastered this concept. So they could build surface antigens that are not degradable by the proteasome anymore. So they actually can stop the proteasome, they can stall it in a certain way, and with this they're invisible to the immune system because no peptides from them are there. So the cells look like normal, healthy cells, even though they're infected by a virus. And we would like to understand, how do those proteins do that? Can we do something against? Can the proteasome help in some way? Because of course there's always co-evolution. So if a virus could find a way of tricking the proteasome to not do anything or stop the proteasome, the cell has a huge benefit and do something against it. And there are definitely mechanisms around, so there's a couple of different proteins that can help to rescue the proteasome from such states, but in the named viruses, there is no way, so those viruses can escape the proteasomal signal, and we don't really know how, but if we would understand how, we probably would get a tool to actually do something against them. The Vienna Biocenter is a very cool place to do science because it enables you, even though you're not mastering every technique, to basically do every technique that you will do, because your colleagues or your PI colleagues definitely can do most of the things, and you can just go to a colleague that masters some things. I was never dreaming that I would do genetically modified organisms or that I could do experiments in cells or in whole organisms. But being here, I figured out very fast that I just have to go to one of my neighbors and say, I would like to do that, and they will help me with everything they can do. So in principle, I'm only limited by ideas, and not even that, because we get lots of insights from each other and we see a lot of things and can basically think out of the box because of lots of possibilities, which is not only helped by the colleagues, but also by many great facilities that help us a lot. For instance, is the workshop of the IMP that has great engineers and great workers that can build machines that we just dream of, so that we make a pencil sketch and can actually do whatever we think. And everyone is very open-minded, and the whole campus is extremely diverse. So there's everything done from ecology to down to like individual molecules, what we are actually doing, and the correlation is that we can do here is amazing. And this is what I really like about the BBC and the IMP especially. So apart from biological groups, there's a lot of institutes in Vienna that focus basically on every kind of research, and we're happy that we can do very technical things that are not generally biology or not considered to be biology, and we can collaborate with people from the Technical University of Vienna or the University of Vienna or the Institute of Science and Technology Austria, with people that are generally doing things like microfluidics or machine learning that have much broader concepts that are not only made for biology but can help biology a lot. And also in these communities we can openly discuss, and everybody in the Vienna area is very open to collaboration, and even people I've never met, I can just send an email and they will help and help with their expertise, which is a great thing. And even the facilities of the other institutes are very open to help people, which gives a great community. and also a very interdisciplinary view of science. So in principle, well, the limit is the sky, basically. So I've not come across any idea that I would like to do where I couldn't find a person in Vienna that could help with that or was even willing to help with that. Basically, everyone I approached was always willing, and this is a very great thing. From the beginning, when I was a small child, I always wanted to study medicine to understand how a human being is working or how something can be a living organism. And this went quite a long time during my school career. And only very much to the end, I decided that medicine will not help me in this, because medicine will not give me the possibility to understand everything in details or in the details that would be sufficient for me. So then I decided to go to more scientific point of view and the possibility to find out things myself, to not only learn how things are working, but also find techniques how I can find out things for myself. So I started studying biochemistry and soon joined in with physics as well to get an understanding how the world actually works in terms of the forces and the energies with biological matter. So what always interests me and which keeps interesting me is how something that is not living, like a small molecule, is forming something that is living. So how is this transition going? And this was what motivated me to study biochemistry and physics at the time. And I think in the very early in my studies, I got very much interested in the crossplay between the techniques and biophysics, because in this way I could really understand for myself in terms of how a molecule really works from a very fundamental point of view, which in my understanding is the way to understand this transition from something that is not living to something that is living. But also in that time I saw a very inspirational talk, so I got the possibility of going to a symposium in my second semester of the German Academy of Sciences, the Leopoldina, and I saw a couple of biophysics talks that basically changed my life. And this was one talk that was particularly interesting, featured cryo-EM and showing the possibilities that I could actually see individual proteins or things like ribosomes that I knew from the lectures. That triggered me so strongly that I really wanted to do that for the rest of my life. And this is what I'm doing since, and I definitely never found anything against that. And I'm so happy that I can actually do what I really love, and I can understand so many of the fundamental questions that are fundamental for me with this technique.